come to the next speaker. That is Professor María José Alonso from Spain, and she will introduce into their strategies about multi-layered nanocapsules for cancer therapy.
Okay, thank you, Mr. Chairman, for the kind introduction. Good afternoon to everyone. Well, thank you for being here at this time of the day. I guess everybody's tired now. So let me see how we move. So, of course, I'm very pleased to be here and to share with you the results of the work that we are doing at the University of Santiago de Compostela in Spain. And for sure, I'd like to thank Professor Loeffler for inviting me to come and present here this work aimed at designing multi-layer. Let me see how the pointer. Okay, good. So our work trying to design multi-layer nanocapsules as carriers for multi-target oncological nanotherapies. So basically, what we are doing in my group in Spain is designing nanocarriers, taking into account different aspects. And in particular, we talk about regional design in the sense that basically our nanocarriers are characterized by having kind of core surrounded by some polymer layers. And the core is defined taking into account for sure the nature of the drug we want to deliver, whereas the coating or this kind of corona we have around is decided taking into account the biological environment and for sure the barriers, the biological barriers that we want to overcome. So based on this, in the area of cancer, we have tried to develop specific nanocarriers for either active or passive targeting, and we call them multi-layer nanocapsules. I will show you now why. But basically, these nanocarriers have been defined taking into account that we want them to go to the tumor tissue. We want them also to go to the lymphatics, so either passive or active targeting to these tissues. And regarding the target cells, we have been trying to target different populations within the tumor and also in the lymphatics, in particular the cancer cells or metastatic cells in the lymphatics, and also the immunosuppressor cells. So basically, we have used until now different pharmacological approaches. So in some cases, chemotherapy, cytotoxic drugs for sure, for killing, for causing the death of cancer cells, but also small amounts, low doses of cytotoxic drugs for achieving immunogenic cell death. So both approaches. With regard to the other immunotherapies, we have tried also to deliver monoclonal antibodies to the cancer cells and also cRNA and microRNA to reprogram or re-educate these immunosuppressive populations. We have also tried to deliver here in these immunosuppressive cells cytotoxic drugs in order to kill them, and also some immunomodulators. So all these strategies are being explored in my lab, and in that case we are trying to, for sure, achieve immunoactivation of these immunosuppressor cells. So all of them are being attempted with this multi-layer nanocapsules delivery platform. And these nanocapsules have been developed while we were working in the LymphoTarg European Consortium. And this activity ended 1 year ago. And within this consortium, we managed to develop some nanocapsules with lympho-targeting properties, with an affinity for the lymphatics. Then this European consortium, this project has been continued with a niche European consortium that started working 1 year ago. And within this consortium, our main goal is dealing with the cancer cells for inducing immunogenic cell death. And right now, we are also starting this worldwide cancer research consortium in collaboration with the group of Professor Mantovani in Milano. And in that particular consortium, our goal is to deal with the tumor-associated macrophages. So many different objectives, several oncological, pharmacological therapies, but using the same kind of delivery strategy, because these nanocapsules are quite versatile. As you can see here, they are composed of a core, in this case an oily core, where we may dissolve a drug, and this core is conveniently stabilized, and then we can coat this core with one layer where we can put another drug if we wish, or another layer which may control the release of the encapsulated drugs, and another layer. I mean, we can put subsequent layers around these oily droplets. So until now, we have seen that it is possible within this system to encapsulate one or more active ingredients, controlling agents, and also targeting myitis. We have linked here to the surface. So something also interesting to mention, although I did not bring the details with me in the slide, but I can share with you, is that these nanocapsules are produced by very simple methodologies. One of them is, for example, by self-emulsifying techniques, so very simple and easy to scale up. So regarding the kind of characterization we have performed until now with these nanocapsules, for example, we have looked at their behavior in these kind of reactors, three-dimensional co-cultures of tumor cells and lymphocytes. And basically what these images are telling us is that the nanocapsules in red are mainly localized, are internalized by the cancer cells, whereas they do not enter the lymphocytes as we expected. We have also, as I said before, we have loaded monoclonal antibodies within these nanocapsules, and we have verified the possibility of these nanocapsules. Despite of their lipophilic character, we were able to associate monoclonal antibodies and to deliver them inside the cancer cells. So what we see here, these green spots represent where the antibody is located. So intracellular delivery of antibodies. And we have also studied the capacity of these nanoparticles, nanocapsules, in fact, to cross epithelia, to cross barriers. And for this, among other approaches, we have used the zebrafish model. So we have administered the nanocapsules, different types of nanocapsules in this zebrafish model. What we see here basically is very illustrative. This is why I brought it here, because we have some kinds of nanocapsules that are able to cross all these epithelia, whereas other types of nanocapsules accumulate in other places. So this is a nice model to see just the capacity of them to cross epithelia. So what I would like to do now is to show you the specific data we have obtained for one of these prototypes. For example, for those loaded with Docetaxel as a model drug. So we have prepared these nanocapsules, and we have seen, as I told you now, that these nanocapsules have the capacity to cross epithelia. They have also shown a selectivity for cancer cells. They are stable in plasma. We have also this data, and we know that they can hold the drug, avoiding premature release. So we have also performed some pharmacokinetic and efficacy PK/PD studies. And for this, we have used this animal model, orthotopic metastatic lung cancer model, as you can see here. So basically, we have injected the cells, and then after 20 days, we have administered a dose of these nanocapsules containing Docetaxel, and a week later, another dose. We have been using either 5 or 10 milligrams per kilogram of Docetaxel, as most people do. And then we have looked at the pharmacokinetics and also at the efficacy, mainly in terms of the number of metastatic cells, not only tumor growth, but also metastatic cells. So what you can see here, these are quite interesting data, because we are representing the percentage of the dose that we found in the tumor tissue. And you see that the amount we found is really high. It's enormous. We have never seen such a great amount of Docetaxel reaching the tumor tissue. And in fact, you see that even after 24 hours, the amount of Docetaxel that is in the tumor is 39 times higher than the one achieved with the commercial formulation, Taxotere. So that proves the targeting capacity of these nanocapsules. And similar results, although the accumulation was lower, could be found in the lymphatics. Again, here you see that Taxotere reaches the lymphatics in a low amount. However, when we administer our nanocapsules, the amount of drug that reached the lymphatics was significantly increased. So then when we look at the efficacy of these formulations, I mean, here we are just showing the luminescence in the tumor over the time, and you see that the targeted nanocapsules allow us to control the size of the tumor. But when we did the quantitative analysis of the luminescence, we found in the tumor tissue, in the lungs, you see that in the case of the control, actually the mice were really bad after 37 days, and we determined the load of cancer cells, whereas in the case of those treated with Taxotere or with our nanocapsules, they survive much longer, and at 47 days, we had this amount of cancer cells in the tumor, and you see much lower amount when they were treated with the nanocapsules. But more important than this, maybe, is the fact that we were able to reduce almost to zero the number of metastatic cells in the lymphatics. And this was not the case for the commercial formulation, as you can see here. So in terms of survival, the animals treated with the nanocapsules survived all over the experiment, even more than 47 days. Most of them were fine, whereas those with Taxotere or non-treated, of course, they died much before that time. So just to summarize, I had to make it simple and brief, but basically this is a very versatile delivery platform consisting of an oily core, several polymer coatings around different coatings and targeted with different ligands. But depending on these formulations, we have achieved either passive targeting to the lymphatics or active targeting to the tumor. And we are also exploring now the possibility of using the same kind of systems for re-educating the myeloid-derived suppressor cells and tumor-associated macrophages, although we are still in an early stage. And we have only performed some in vitro studies showing that it is possible to re-educate these suppressor cells. And this is all right now. I think the 15 minutes are over. And for sure, I'd like to thank my group of research at the University of Santiago de Compostela and the financial support we have obtained from the European Commission through these different consortia I mentioned to you before. Thank you very much for your attention.
Yeah, thank you very much for sharing this interesting approach with us. Other questions from the audience? I may have one, maybe you mentioned it in your presentation, but you were speaking about active targeting. What did you use as the target and as the ligand?
Well, we have been using peptides more than monoclonal antibodies, different kinds of peptides until now, depending on the target cell.
Mm-hmm. Is among them transferrin receptor targeting peptides?
No.
Oops.
Mike,
I mean, the targeting was truly impressive. Can you comment on that?
Well, you mean the accumulation in the tumor tissue? The only, I cannot comment, sorry, the type of ligand we have used in that case. So with the passive targeting, we have achieved a significant accumulation, but as you could see, much higher with the with the targeted ones. Yeah, you mean this, right? Yes. Okay, so we think there is mainly a tumor targeting, and we think that this high concentration is due to the tumor targeting strategy, but also to the penetration capacity of these nanocapsules. So I can tell you that we have been using polymers which enhance the penetration capacity as well. So we don't think it's only due to the peptide ligand, but also to the polymers that we are using to enhance the penetration and transport of the nanocarrier through the stroma. Mm-hmm.
I just want to pick up on this a little bit further. When you looked at the lung tissue, did you see that the lung tissue was in really bad shape because you had such good targeting and such high kill rates?
No, actually, when we did the anatomopathological analysis of the tissue, I mean, those treated with this very high concentration were the appearance was really good. So, and they survived all over the experiment. Mm-hmm. In one case, 80% of the animals survived. In the other case, 100% for 47 days. What we are doing now, though, we are trying to complete this to better understand this data and to see what the consequences are. And we are also looking at the capacity of these nanocapsules to kill the tumor-associated macrophages. This is where we are now. But these data were obtained a month ago. So we are still trying to elucidate.
Very
but
impressive.
The pharmacokinetic analysis was very rigorously done, I mean, by LCMS and validated, and I mean, it's not imaging, it's drug concentration.
Yeah, yeah. Did you, you also mentioned when you were looking at the liver, you mentioned Kupffer cells. Did I hear you correctly that you said that you saw the particles in the Kupffer cells in the liver? Or did I miss that?
I didn't say that, but it is certainly true. I didn't bring the whole biodistribution we have. It is certainly true that we found some amount of Docetaxel in the liver as well. But it was the same amount that we found, or a bit lower, that we found with the Taxotere. So there was not specific accumulation due to the nanocapsules.
Okay, there was one question which became obsolete in the meantime. Or?
It's the same question that he asked. The accumulation in liver and spleen.
Yeah. Well, I have the slide with me. I can show you later.
Okay.
But it's the same as the non-encapsulated Docetaxel. There is not a specific accumulation in the liver, no.
Okay, then thank you again for showing us the power